endogenous cannabinoids today and their receptors. Hopefully, by the end of the presentation, you'll have a better understanding of where exactly the THC has action in your body. All right, so first of all, endocannabinoids, endo, something Greek, meaning within. Cannabin, referring to the cannabis sativa, which the original action of this receptor was deduced from. You might know it as hemp or marijuana. And oid, meaning like that of, so in the end, cannabis, like substance within. So we're not going to be talking about THC in this presentation, we're going to be talking about the things that we have named after the THC. Or the cannabinoids. All hail the plant. Dietary sources of endocannabinoid precursors are mainly omega-3 and omega-6 fatty acids. You might have heard a lot about these things in fish oils and various seeds, flax, hemp. I know Alex mentioned them in the process acetaminophen. That's actually also used in the synthesis of prostaglandins, among other lipid signaling devices here. Here's arachidonic acid, which he made reference to as well, and it is the precursor fatty acid to anandamide, which comes from a Sanskrit word meaning bliss, and is one of the endogenous cannabinoids that has been isolated from the human body. Here's a few more examples. But basically, they have two main features. They have this long, unsaturated fatty acid chain, or motif, and also a polar head group, often consisting of an amine or a glycerol, or sorry, an ethylamide, or this glycerol group. Interesting thing to note about endocannabinoids is they are produced on demand and are very rapidly metabolized. So they are short-range devices that are responsive to other transmissions being done within the body. So in the cannabinoid 1 receptor often is used to down-regulate neurotransmitter release. It's produced by the. Sorry, back to this. Yeah, and so these things they're rapidly formed because they're actually stored in the lipid bilayer. This phosphatidyl phosphatidylcholine molecule is actually, if one of these was the arachidonic acid fatty chain molecule, that would be like a way, on the path to developing the endocannabinoids. So because these things are found in the membrane, it's sort of not under the same mechanisms as other neurotransmitters which act upon exocytosis through vesicles or vesicles. So we have our dietary source of arachidonic acid here, fatty acid, and it will carboxyl group will interact with the amine of phosphatidylethynolamine to form this beautiful molecule here, which will be in the membrane. And when we need the endocannabinoid release, it'll be cleaved at the phosphate group and form our endocannabinoid, or anandamide. and then subsequently metabolized fairly quickly by fatty acid amine hydrolase and I believe this is acetylglycerol, thank you. And again, just wanted to take note at the I'm missing a molecule in here, but if you cleave this here, it's pretty much a phosphatidylglycan or phosphatidylcholine, like molecule. So there's two basic types of cannabinoid receptors as far as the current classification goes. The first one was discovered in the brain, which mediates most of the behavioral actions, sort of the getting high actions of the cannabinoids. Like obviously you don't necessarily get high while you are endogenously producing them, but I guess some studies have been done, I should note, where you either knock out some of the degradative enzymes, which lead to enhanced accumulation of endocannabinoids and altered behavior. It's interesting to note that it is the most abundant G-coupled protein in the brain. I think the study was actually undergone on rats, but I don't know if it's been done on humans. It's interesting to note. The second type is the CB2, the cannabinoid 2 receptors, originally isolated from spleen tissue, and previously, again, in the past referred to as the peripheral cannabinoid receptor, but it's found throughout the body and even in brain tissue, especially in immune cells. And when you have an immune response, you're likely to see expression of CB2 receptors. And again, both receptors are G-coupled protein receptors, which, as most of you are probably familiar with G-coupled protein receptors, upon binding of the ligand, or the endocannabinoid, to the exterior of the cell and of the receptor, mediates a change which releases this G protein, cleaves it into its alpha subunit and beta gamma subunits, and they have different actions depending on what type of variant they are. And I believe there is some variation in how they are expressed. Typical activation of a cannabinoid receptor will often result in the closure of calcium channels, which is a major cell signaler. When calcium channels reach a high enough point, you'll often see increased activity in the cell, as well as things like exocytosis of neurotransmitters, so releasing of neurotransmitters. Opening of potassium channels also occurs, which in turn prevents the calcium channels from opening and being released, or the calcium from being released. And then inhibition of adenylene cyclase, which lowers cyclic AMP levels and reduces the action of some protein kinases. So in a typical neuron, presynaptic neuron, when you have a pulse or a voltage, this firing of the neuron, you'll have a flux come down, whatever. It'll cause a shift of, or it'll cause the calcium channel to be opened, and calcium will flood into the cell, which will trigger the release of the neurotransmitter, which will travel to which will be received by the postsynaptic neuron and increase the calcium level in that neuron, which will in turn, which will in turn activate endocannabinoid production, which will find their way back to the presynaptic cannabinoid receptor, which will reduce the amount of calcium in the cell, open the potassium channels, and reduce the cyclic AMP, slowing the release of neurotransmitters to the postsynaptic neuron. So to review again, the increase in the charge of the presynaptic neuron reaches a point such that the calcium rushes into the neuron and triggers the exocytosis of your neurotransmitter, whether it be glutamate or GABA or serotonin, norepinephrine, which is received by the postsynaptic neuron. In this case, it's a glutamate receptor, I believe, or AMPA receptor.
That's correct. It's one type of glutamate.
Yeah. Which then, with significant activation of the postsynaptic neuron, generates the cannabinoid, which goes back and in a retrograde transmission dampens the release of the neurotransmitter from the first membrane.